Now I am going to discuss about the famous Watson and Crick's model for DNA structure, which is commonly called as DNA double helix. In 1953, James D. Watson and Francis H.C. Crick. James D. Watson and Francis H.C. Crick proposed a model for DNA structure. In 1953, they had proposed a model for physical and chemical structure of DNA molecule. The model they devised, which fit all the known data on the composition of the DNA molecule, is the now famous DNA double helix model. At the time of Watson and Crick's work, DNA was known to be composed of nucleotides, but it was not known how the nucleotides form the structure of DNA. The data they used to generate their model came primarily from base composition studies conducted by Chargaff and X-ray diffraction studies. This one was performed by, these base composition studies are performed by Chargaff and X-ray diffraction studies. This X-ray diffraction study, Rosalind Franklin and Horace H.F. Wilkins. So, prior to discussing about the Watson and Crick model of DNA double helix, we should concentrate about the base composition studies by Chargaff. What was the study of Chargaff? Chargaff hydrolyzed the DNA of a number of organisms and quantified the purines and pyrimidines released. His studies showed that 50% of the bases are purine and 50% of the bases are pyrimidine. 50% bases are purines, 50% bases are pyrimidines. Okay, and amount of adenine equals to amount of thymine. Amount of adenine equals to amount of thymine and amount of guanine equals to amount of cytosine. These equivalences have known as Chargaff'S rule. In comparisons of DNA from different organisms, it is known that the ratio of A by T is 1 and ratio of G by C, ratio of A and T is 1 and ratio of G and C is also 1. But A plus T, ratio of A plus T and G plus C, it's not equal to 1. It is called, and this one it is called base ratio. This one is called base ratio. Ratio of A plus T and G plus C. This is not equals to 1 and A plus T, G plus C ratio is less than 1 in case of prokaryotes, as the DNA of prokaryotes are GC type. GC type. This ratio is greater than 1 in case of eukaryotes. As we know, the DNA of eukaryotes are AT type, means the amount of AT is greater than amount of GC, and here in prokaryotes the amount of GC is greater than amount of AT. So AT-GC ratio in prokaryotes is less than 1, and AT-GC ratio A plus T and G plus C ratio in eukaryotes is greater than 1. And this one is known as Chargaff'S rule and base composition, and this is the whole findings of base composition studies of Erwin Chargaff.